Okay, let's get started. So thanks for everyone for coming to this special rapid fire session. So before we start the session, I just wanted to let everyone know that Bruce Stillman and I have made a call and we've decided we'll have the picnic down at the lab beach. It's a little iffy, the weather, so we hope it'll work out. But I guess we should just get started with the session now. So I'd like to introduce my co-chair, Yan Li, from the Tianxiao and Chrissy Chen Institute, who are supporting the symposium. And just to explain how we selected the Chen Travel Fellowships. So we put out calls for applications and we received about 45 applicants. And here at Cold Spring Harbor, we had a small committee who read through those great applications from everyone. And we triaged those down to around 20 applications, which we forwarded on to the Chen Institute, and they made the final decision. So these are short talks by young scientists, and they're 2 minutes in length. No time for Q&A, but if you do have questions afterwards, they're all giving posters in this afternoon's poster session. So I guess we should begin. Do you have anything to? Sorry. So the speakers were all awarded travel fellowships from the Chen Institute. I should make that clear. So that they got a dispensation to basically come to the symposium at no cost, which was a terrific contribution from the Chen Institute. So, okay, we're going to pass on to.
Our first speaker this afternoon is Kun Li from Rockefeller University.
Okay, thanks Tianjia and the Christian Foundation for this generous fellowship. And we are very interested in understanding why the frequency of human social and emotional disorders varies significantly between male and females. Taking advantage of trap technique, which has been well introduced by Dr. Yang Dan yesterday, we identified a specific class of oxytocin receptor interneurons in the mouse prefrontal cortex. And we found that modulation the same population of oxytocin receptor interneurons in the medial prefrontal cortex could generate sexually bivalent social and emotional disorders. And to get a better insight into the mechanism, we profile all the mRNAs translated only in this specific cell type. And quite interesting that we have identified a specific class of a specific protein in this cell type. It's called the CRHBP. And here, this is just showing our model. We found that we think there are two pathways in the medial prefrontal cortex coordinated by oxytocin receptor interneurons. In females, oxytocin receptor interneurons employ a GABA-mediated inhibition on layer 5 pyramidal cells to generate a prosocial behavior, while in males, CRHBP released by oxytocin receptor interneurons is sufficient to bind with CRH and suppress the activation of CRHR1 receptors on layer 2-3 pyramidal cells and cause an anxiolytic effect. And our idea is that the local action of oxytocin and CRH in the medial prefrontal cortex, coordinated by oxytocin receptor interneurons, may be involved in the modulation of a wide variety of human behaviors. So if you want to know more details about our model, please come to my poster and talk with me. Thank you.
Our next speaker is Xiaodong Li from Nagoya University, Japan.
Hello, I'm Xiaodong Li from Nagoya University, Japan. Everyone here was not born with the language skill, right? You learned it. So in case of Drosophila, for a very long time, people believe that the most processes in the cognitive behavior of Drosophila are innate. However, in my study, the results support that the flies learn the sound discrimination. And here is the phenomenon. We use the sound, cognitive sound playback to train the flies. With the training of the conspective sound, the flies, both male and females, significantly reduce the response to the heterospecific sound. However, no training of heterospecific sound training cannot lead this learning. And further, I found GABA signaling is necessary for this auditory learning, and it functions on GABA A receptors in a small group of neurons called PC1 to mediate this learning. So our model is that beside this innate auditory circuit for the cognitive sound processing, the auditory experience can recruit the GABAergic inhibition to modulate the cognitive choice. So, this experience dependent plasticity shares many similarities with the auditory learning in songbirds and humans. And now I'm investigating whether there is a critical period for this and how the memory is modulated in this process. And please come to my poster if you want to know more details. And now this year I will graduate. If you want to find a postdoc, I have my own funding. Please talk to me. Thank you.
Our next speaker, Shawn Liu from Whitehead Institute, Boston.
So first, thanks to the select committee to select me for this Chen Fellowship. What I study is epigenome, so we call it the brain of the cells. So the reason is it can really integrate the signal both from environmental and developmental cues to encode the cellular memory in the format of chemical modifications occurred at the chromatin. So to understand the function of those epigenetic events, so we developed the tool, particularly to understand methylation, we developed the tool by fusing catalytically inactive CRISPR-Cas9 with the demethylation modifier enzyme TET or DMT to mediate demethylation editing. So with application of this tool, we studied the hypermethylation events at the CGG repeat, which at the FR1 locus in which we know silence of this gene called Fragile X syndrome, the most common inheritable form of intellectual disability in males. So we find out once we demethylate the CGG repeats, we can reactivate these genes and we can rescue the hyperactivity phenotypes associated with the Fragile X neurons. So moving forward, I hope I plan to systematically characterize the epigenetic events accompanying the brain functions such as learning and memory. Then we can dissect the cellular functions. Hopefully, eventually, we have a better understanding of contribution for the epigenome to the brain functions. I have a poster too, and welcome to stop by my poster. Thank you.
Next speaker, so Yoav Livneh from Harvard.
Okay, so first I'd like to thank the Chen Institute also for their support and correct a mistake in the program that only my name is listed, and I of course do not work in isolation. I'm actually a postdoc in two great labs, sort of Brad Lowell and Mark Anderman. And in my postdoc, I've become interested in the question, the very broad question of how the state of the body affects sensory processing, or maybe put even more broadly, how it affects perception of the world around us, right? And you can think of an easy example here on top is you're watching a fruit commercial and you're gonna have different responses to that, whether you're hungry or sated, or a beverage commercial, whether you're thirsty or not. And this is obviously a very complicated question, but we decided to sort of break it down into two components. On one end, we have cortical processing of these predictive cues and what they mean. And on the other end, we have sensing of the internal state of the body, which is performed, among others, by hypothalamic neurons, distinct populations of neurons that are sensitive to things like your blood glucose levels, your blood sodium levels, various hormones from your digestive system. And we wanted to know how these two things interact. So on the cortical side, we focused on insular cortex, which has been, at least by human literature, implicated in such things. And we developed a new experimental approach to image the activity of insular cortex neurons over long periods of time through a microprism. And then we combined this with cell-type specific manipulations and circuit mapping of specific hypothalamic neurons, artificially creating states of thirst and hunger in behaving mice. And sort of the summary at the bottom here of our model, what we've found is that distinct neurons driving either thirst or hunger actually converge on a common pathway in the limbic system, specifically through midline thalamus and amygdala to modulate cortical activity. And we speculate that this is not only a set of convergence for thirst and hunger, but at least the anatomy suggests it's actually a set of convergence for other need states, such as temperature seeking, warmth seeking when you're cold, and maybe the drive to copulate, and any other things like parental behavior. So please come to my poster. I'll be happy to talk about it more. Thank you.
Next speaker, David Ottenheimer from Johns Hopkins.
Thank you, and thank you for this honor. Hi, everyone. I'm a graduate student in Patricia Janik's lab at Johns Hopkins, and our lab is interested in the neural circuits underlying motivated behaviors. In particular, I am interested in how the brain processes highly palatable rewards. So say, for instance, you were to reach for this glass of mysterious orange liquid on the left side of the screen. It may be orange juice or mango juice, and if you're like me, both are pretty good options, but when you take that sip, what the juice ends up being might affect how quickly you drink it, how quickly you take your next sip, how big each gulp is. That's the kind of processing that I'm interested in studying. So to model this in rodents, I've designed a task where a white noise cue announces to the rat that they will either get sucrose or maltodextrin on that trial. And these are two highly palatable carbohydrates with equivalent caloric value. So I can then go into the brain and see whether there are traces of information about these two rewards in these regions, the nucleus accumbens and ventral pallidum, that are known to be involved in reward-seeking behavior. So surprisingly, I've found during my recordings, as I've color-coded here, there are many more neurons in ventral pallidum that actually have traces of reward-specific information about sucrose and maltodextrin. And this information is coming earlier in ventral pallidum than it is in nucleus accumbens. And if you remember, as we've been talking about these regions today, most people talk about ventral pallidum as an output of nucleus accumbens. So if you want to talk to me more about why it might be that ventral pallidum is representing this information earlier than nucleus accumbens, and some of the techniques that I've used to get at these answers, like linear discriminant analysis, I would love to talk to you more at my poster. Thanks.
Next speaker, Jing Ren from Stanford.
Good afternoon, everyone. I'm Jing from Lichun Law's lab. Serotonin system is one of the most mysterious and complicated system, and we are focusing on the R-serotonin system, which is the major serotonin resource for the forebrain. And it has been implicated that the R-serotonin system is involved in multiple mood disorders, including depression, anxiety, aggression, and so on. But this DR-serotonin system have been mostly studied as a unitary system. So we here reported that this system has multiple parallel pathways. And we used a viral tool to dissect it based on its downstream targeting brain areas. We found that the serotonin neurons projecting to OFC and the serotonin neurons projecting to CA received largely biased input from specific brain areas. We also found that these two groups of neurons have largely complementary collateralization output. Moreover, we found that when the animals receive reward, both of these two groups of neurons behave activation, as showed here. But when the animals receive punishment, the serotonin neurons projecting to OFC was inhibited, but the serotonin neurons projecting to CA was activated. In addition to this, we also used the gain of function and the loss of function behavior task and find out that the activation and inhibition of these two systems have different influence on the anxiety-like behavior and depression-like behavior. So if you want to learn more about this story, please come to my poster, and thank you very much for the fellowship.
So our next speaker, Prerana Shrestha from NYU.
Hi, I'm here to tell you about a tool that I have developed with a former student of Nat Hines, Pinar Ayada, and I'm currently a postdoc in Eric Klein's lab in NYU. So this tool, we've named it CYPSI for cell type specific, preconditional, and drug-inducible protein synthesis inhibitor. We have used this tool to interrogate cell type specific translation in long-term threat memories. So the actuator molecule in our system is a kinase for EIF2 alpha, which is a key initiator translation initiation factor. So phosphorylation of EIF2 alpha converts it into a competitive inhibitor of EIF2B and stops the translation machinery at the initiation step. So we have combined the PKR kinase, which is the kinase for EIF2 alpha, with a protease that is derived from hepatitis C virus, which targets the IPKR in steady state. But when we introduce a drug that blocks the protease, now we release the kinase into the system and block general protein synthesis. So we've shown you can come to my poster to see the data, but we see a 60 to 80% decrease in general translation. And when we apply this system to pan-neuronal CYPSI nestin creline, we see a really robust decrease in long-term memory expression and consolidation, tested 24 hours later after auditory fear conditioning. We also find that there is a sensitive time period after learning, so there's only a 1 hour of time period following training that the memory, long-term memory is sensitive to disruption with protein synthesis inhibitor. We further have applied this approach to CAMK2 alpha expressing principal excitatory neurons in lateral amygdala as well as in central amygdala somatostatin neurons. And we find that these both neuron types have very distinct roles in long-term memory consolidation. And we tried reactivating the CAMK2 cells, CAMK2 alpha cells, the following day with DREAD receptor activation. And we find that while we can recover part of the lost memory, the precision of the memory is actually still lost in these mice. With somatostatin mice, what we have seen is that there's no loss in discrimination index towards a paired tone versus an unpaired tone, but they are really impaired in responding to the paired tone, so their long-term memory is impaired. So this is a tool that can be applied to a wide variety of biological processes, including memory, drug addiction, as well as imprinting, for example. So really, these are examples of how we can apply CYPSI. It's really a resource for blocking de novo protein synthesis in a specific cell type manner, in a drug-inducible manner. Thank you.
Our next speaker, Ben Yang from Northwestern University.
Hi everyone, my name is Ben from Jim Sirma's lab at Northwestern University. And I want to first thank the Chen Institute. So how emotional states such as fear affect need states such as feeding is not well understood. Loss of weight and appetite has been reported in PTSD patients, other stress disorders, as well as inferior condition animals treated with other stressors. So here we use fear conditioning as an animal model, and we focus on 3 nuclei, the central nucleus amygdala and the locus ceruleus, both of which play pivotal roles in fear conditioning. And the peribrachial nucleus, as you heard from yesterday's talk, is important for arousal, but also plays important roles in feeding. So we first found that central nucleus amygdala has inhibitory input to peribrachial nucleus, but not to the locus ceruleus. And then we found reciprocal excitatory condition between the locus ceruleus and the peribrachial nucleus. We further found that amygdala input to peribrachial nucleus is depressed after fear conditioning. Now let's zoom in to see the mechanism. The mechanism of depression is induced by endocannabinoid release and activation on CB1 receptors on amygdala terminals. And we further found that upon fear stimulation, locus ceruleus releases both norepinephrine and glutamate, which activate alpha-1 general receptors and type 1 mGluRs respectively to induce endocannabinoid synthesis and release to mediate heterosynaptic LTD. Activation of PBN is known to terminate feeding, and indeed we found fear-conditioned animals ate less than control animals. And if we knock out CB1 receptor, specifically in the central nucleus amygdala, to block LTD, we block reduction of food consumption. So together, we found a disynaptic input from amygdala to locus coeruleus via periapical nucleus and reciprocal connection between the locus coeruleus and periapical nucleus. And we found amygdala input to periapical nucleus is depressed after conditioning, and the mechanism of the locus coeruleus induced endocrine synthesized mediated heterosynaptic LTD. And finally, we found fear reduced food consumption, which can be blocked by knocking out CB1 receptors, specifically in central nucleus amygdala, to block LTD. My poster number is 87, and I'll be happy to talk more about this. Thank you.
Our last speaker for this session is Zhe Yang from Zhejiang University, China.
As we know, mammalian systems relies on umami system to evaluate protein diet and promote feeding. Fruit flies, however, they lack the homolog or umami receptors, and they must employ distinct amino acid sensing mechanisms by measuring the volume of ingestive or individual flies. We found that flies can tell the presence of amino acids and promote feeding within the time scale of about 20 seconds. We found that not gustatory systems, but 6 diuretic hormone 44 positive neurons in the central brain are necessary and sufficient for amino acid to promote feeding. We further found that 3 amino acid out of 20 amino acid, glutamate, alanine, and aspartate are necessary necessary to promote feeding and activate those diuretic hormone 44 positive neurons. In short, upon feeding, glutamate, alanine, and aspartate are swallowed into the gut and activate diuretic hormone 44 positive neurons by their amino acid transporter CG138 to promote feeding and result in the homeostasis of protein. Thank you.
Okay, thanks very much. So that was a great session. Everyone has posted the session, so let's just thank all our speakers again.